Welcome to the problem set for the eukaryotic chromosome review. Question 1. Using the word bank below, choose the element that each statement describes. So for A, an individual's complete set of chromosomes. That is otherwise known as a karyotype. And that's why the term karyotyping is where they take somebody's DNA and look at the complete set of chromosomes that they have, and that can see chromosome aberrations like Down syndrome, Turner syndrome, if it's XO, and you can also see if an individual is male or female based on the sex chromosomes you see in that karyotype. B. Shortens with every cell division in a normal somatic cell. So this is a telomere, right? And the telomeres are the ends of the chromosomes, and these shorten in what's known as the end replication problem because the RNA primer that DNA polymerase needs to use gets removed, and then you have this 3' overhang and a 5' truncated strand, and that's why the telomeres shorten with every cell division. C. Complex of DNA and proteins in the nucleus during interphase, which is not visible under the microscope. So this is just general chromatin. And when chromatin isn't compacted into that highly compact radioscaffolding, you can't see it under the microscope, and it's not those chromosomes that we normally draw. So it's not, looking like this, which are the chromosomes we normally draw, and you can't visualize them under the microscope. D. Type of chromatin that is not densely stained and contains protein coding genes. So this is just, again, this is application of theory and vocabulary terms, but this is what's known as euchromatin. Okay, so just know that euchromatin contains the protein coding genes. E. Proteins titled H2A, H2B, H3, and H4. So these are what's known as the core histone proteins. So remember, these proteins are present in the octamer of the core nucleosome, and they're each present in 2 copies in each nucleosome. Okay. Part F. About a third of chromatin composition, and this is largely acidic. So this is just, again, something you need to know from the theory. These are what's known as non-histone proteins. So the non-histone proteins make up about a third of the chromatin, and they're largely acidic. Part G. Site of attachment of kinetochores. So this is the centromere, right? It's an important DNA sequence element. And this is the element of the chromosome where the kinetochores attach, and therefore the chromosomes can be pulled apart during mitosis and meiosis. Part H. First level of compaction in which DNA double helix spools around an octamer of core histone proteins. So again, as we said before with Part E, this is an octamer of core histone proteins, 4 different types, and this is what's known as a nucleosome. Part I. Densely stained chromatin that generally signifies inactive regions. So this is what's known as heterochromatin. Okay, so just to compare, that euchromatin contains the protein coding regions which are actively expressed. Heterochromatin generally has the inactive regions that aren't expressed. Okay, and then finally, Part J is about a third of chromatin composition, largely basic, and does not contain DNA. So again, these are just the histones, and the histones are just a group of protein that makes up about a third of the chromatin composition. Problem 2. Why does DNA exist in different structural forms at different stages of the cell cycle? So let's run through each of these statements and see which are true, and which explain why DNA exists in different states of compaction at the different cell cycle stages. So for A, chromatin needs to be decondensed in order for DNA replication to occur. So A is true. It needs to be decondensed so that DNA polymerase can come in and actually replicate the DNA before the cell cycle can occur. Before mitosis or meiosis can occur, DNA needs to be replicated so that there's 2 copies. So A is true. I'll just say needs to be decondensed for DNA polymerase to work. Okay, statement B. When DNA is being transcribed, the transcript machinery needs access to open strands. So again, this is the same thing, only this time RNA polymerase needs to be able to get into open strands, so it needs to be decondensed for RNA polymerase. Okay, and just remember that transcription is where DNA is copied into mRNA or RNA transcripts that are then translated into proteins. And then finally C, the genetic information needs to be compact and condensed during cell division. So this is also true, and we've talked about it before in an earlier problem-solving set, that in order for chromosomes to, neatly come apart and separate into daughter cells either in mitosis or meiosis, they need to be compact. Otherwise, it's just this long, not very compact DNA chromatin structure that is really hard to pull apart. So the more compact the package is, the easier it is to pull apart. So during cell division, the chromosomes are in that radial scaffolding compaction state. So chromosomes in radial scaffolding. And remember, that's about 10,000 times more compact than just naked DNA. And that's necessary for the movement of chromosomes into daughter cells, so necessary for movement. So the answer to this question, obviously, is D, all of the above, because all of these statements do explain why DNA exists in these different structural chromosome forms at different stages of the cell cycle. Let's do some true-false on the eukaryotic chromosome. Statement 1. Histones interact with DNA to form nucleosomes. So this is obviously true. And remember that the nucleosome core is an octamer, meaning there's 8 subunits, and there's 4 core histones that are found twice in that octameric core. So these enzymes, these histone proteins, are H2A, H2B, H3, and H4. 2. Telomeres exist at the end of linear prokaryotic chromosome. So this is false because it says prokaryotic, and really it is eukaryotic. So the eukaryotes are the ones that have that end replication problem where you have a truncated 5' end that without telomerase, the enzyme that fixes this, eventually you would get a decrease in chromosome length every time you do cell division. Okay, number 3. 30 nanometers is equivalent to 300 angstroms. So this is true, and nanometers are 10 to the negative 9th, whereas angstroms, which is this symbol here, is 10 to the negative 10th. So 30 nanometers is equivalent to 300 angstroms. And that's important because some professors like to give things in nanometers and others like to give them in angstroms. So it's important to know how to go between the two. 4. Human telomeres contain the sequence TTAGGG repeated multiple times. Now this is true. This is a sequence that they add many times through translocation and then add it to the 3' overhang so that DNA polymerase can come in and extend that telomere sequence at the end of each cell division cycle. Number 5. The DNA in a cell is always tightly wrapped up in the form of a chromosome. So this is false, right? They're only tightly compacted in the form of a chromosome during active cell division. And that's because this compaction is needed for the chromosomes to move efficiently into the daughter cells. division. But otherwise, when they're not undergoing active cell division, like during interphase, they're actually not in the form of a chromosome, they're just in the form of chromatin, which is usually the 30 nanometer fiber. Number 6. Telomerase is a reverse transcriptase that makes RNA from a DNA template. Again, this is another false statement. Remember that telomerase enzyme has an inherent RNA template that it uses to make DNA. So it's just that they flipped these two words. Okay? And finally, number 7, as much as half of the eukaryotic genome may be a repetitive sequence. So this number is true. It says as much as half, so 50% of the eukaryotic genome may be repetitive. So how many copies of each gene is a variable, but this is much different than prokaryotes, where the gene sequences tend to only occur once in the genome. But for instance, eukaryotic, like the human genome, contains at least 50% repeated DNA. So this is true, and this concludes the chapter on the eukaryotic chromosome.